Hello everyone. This video is in continuation with the previous videos on biomolecules, and this video will give us a deeper insight into one of the biomolecules, which is the proteins. So let's start with that. So here, as we can see, biomolecules. The biomolecules can be basically classified into proteins, carbohydrates, fats that we call as lipids, or nucleic acids. Nucleic acids are DNA in the higher organisms and RNA in lower organisms like the viruses, but carbohydrates, fats, and nucleic acids will be taking up in the later classes. In today's class, we'll be discussing proteins, majorly the proteins. Now, what is a protein basically? A protein is basically a main structural component of the cell, which is majorly made up of carbon, hydrogen, oxygen, nitrogen, and there can also be traces of phosphorus and sulfur in a few proteins. So, chemically, the proteins can be defined as polymers of the amino acids or the polypeptide chains. So now let's take up amino acids and the polypeptide chains. So first of all, we'll be discussing about the amino acids. So amino acids. Amino acids are the building blocks of proteins, and there are approximately 20 amino acids that are known to us in nature, essential and non-essential. We have total of 20 amino acids. Now, what is the basic structure of an amino acid? Let's go through it. The amino acids have a carbon compound, have a carbon, alpha carbon. Then this carbon has four bonds. It has a hydrogen attached to one end. It has a carboxylic group attached to another end. It has an amide group attached to the third end. And it has a variable R group. Now, this R group, this variable R group, is basically something that differentiates one amino acid from another. For example, we have an amino acid 1, we have amino acid 2, we have amino acid 3. So the basic structural difference between them is because of this variable R group. So most of the amino acids will have a similar amide group, similar carboxylic group, a similar hydrogen group. This may vary sometimes, but very less times. Most of the times, in fact, every time this R variable group is the group that is responsible for determining the characteristics of these amino acids. Okay, so these amino acids basically join together to form a polypeptide or a protein chain. Now, how are these joined? These are joined by a bond called as the peptide bond. So later on in this lecture, we'll be taking up the structure of the peptide bond. But before that, I would like you to see this structure of amino acid. So here we have the structure of amino acid that I had just drawn. This is the alpha carbon, as we can see. On its one bond, we have hydrogen. Another one is this variable R group. Now the COOH, as you can see, is the carboxylic group, and this NH2 is the amide group. So now moving on, we'll go through this bond, which is called as the peptide bond. So this is the structure of a basic amino acid. Now we'll go to the peptide bond. So what is a peptide bond? A peptide bond is a bond that will join two amino acids. And when two amino acids are joined together, they will form a protein chain. Like one amino acid joins to another, and furthermore, bonds will be joined, and finally, we'll get a protein chain. So, amino acid 1, amino acid 2, amino acid 3, and so on. So, these are joined by the peptide bonds. Now, let's see the structure of this peptide bond. What happens in a peptide bond is that we'll take two different amino acids. So, for example, this is amino acid 1. We have COOH and an amide group. Similarly, this is carbon 2, which signifies that this is basically another amino acid. This 2 will have a carboxylic end and an amide end. Now, to form a peptide bond, these amino acids have to go through a reaction which is called as a dehydration reaction. Now, what is a dehydration reaction? A dehydration reaction is a reaction which undergoes loss of a water molecule. Okay, so now how water molecule will be lost through these amino acids? One of the amino acids will give hydrogen, will take out one hydrogen from here and an OH from here. Now these two will come out and these will give rise to loss of water molecule. Now what is left behind? We can see CO from carbon 1 and NH from carbon 2. Now this CONH bond is called as the peptide bond. Now, as we can see, water molecule is lost. Due to that, the CO and NH will bind together, and because of this bond, this peptide bond, both the amino acids will join together and form a polypeptide chain. They'll form a peptide chain, and further, when more amino acids will be joined, it will lead to the formation of a polypeptide chain. So here we can give a look. We can see this polypeptide chain here. If you can see, this is the amino acid 1, this is amino acid 2. Here you can see H2 and O. One H is lost from the amide group. Another amino acid will give OH. So the loss of this H2O will lead to the formation of a bond between these two amino acids. Here you can see the bond. Now we can see both the amino acids are linked together and they form one a complete structure called as the dipeptide. These dipeptides are joined by the peptide bond, and here we can see that there is a loss of water. So this loss of water signifies that this is a dehydration reaction. So now coming back to our topic, we have covered amino acid, we have covered peptide bonds. Now let's start with the configuration or the structure of proteins. Structure of proteins. The proteins can be classified into four major structures. That is the primary structure. We have secondary structure. We have tertiary structure. And then we have a quaternary structure. So the difference is that basically all these four structures are only formed of the polypeptide chain. But the only difference is that uh, that these polypeptide chains are bound by different kinds of bonds to give rise to different structures. Primary structure is the simplest polypeptide chain. Then we have some complexities in secondary, then some more complexities in the tertiary, and the most complex structure is the quaternary structure. So now let's take up one by one. The primary structure is basically the linear structure like this. This structure basically can be found in the ribosomes. As we know that ribosomes are called as the protein factories of the cells. So when ribosomes form the protein, it is in the form of the polypeptide linear chain, which is called as the primary structure. Now, when we come to the secondary structure, like after translation, for example, when ribosomes have formed the polypeptide chain, now these proteins will have to go through translation. So when they have to go through translation, they are converted to the secondary structures. The secondary structure can be either an alpha helical structure or it can be a beta pleated structure. An alpha helical structure looks like this, and a beta pleated structure may look something like this. Now, this is also a polypeptide chain, and this is also a polypeptide chain. The only difference is in their conformation, because these are joined by different kinds of hydrogen bonds. Due to that, they have a very different structure. Now, because of some forces, because of the ionic bonds, because of the disulfide bridges, this secondary structure will then form a globular shaped structure, which is called as a tertiary structure. So the tertiary structure is formed when the secondary structure undergoes foldings many times because of the presence of ionic or disulfide bridges. So the structure formed as a result of that is called as the tertiary structure. Now coming back to our quaternary structure, what happens in the quaternary structure is that what will happen in the quaternary structure, that this uh, tertiary structure will further undergo some more changes. What will it undergo? It will undergo some hydrophobic interactions. Because of these hydrophobic interactions, the structure will further fold on more, and it will lead to a structure called as the quaternary structure of proteins. And the example of that is hemoglobin. Hemoglobin is the most important quaternary protein that is found inside the human body. Now, if we have to get a better image of this, let's see it here. We have the primary protein structure. So if you can see here, it is the linear chain. So this is the kind of structure that is found in the ribosomes. So once the ribosomes are done with protein formation, it will further undergo translation, and in translation the secondary protein structure will undergo some local foldings because of the presence of hydrogen bonds. So it will lead to either the formation of a alpha helical structure or the beta plated structure. Now, these will further undergo some foldings in the form of a globular shaped structure because of the presence of ionic bonds and disulfide bridges, leading to the formation of tertiary structure. Now, this tertiary structure again undergoes hydrophobic interactions to form the quaternary protein. So, the last thing we have to study here is the function of protein. The protein functions are, because we know protein is present in the human body, in animal body, in any life form that we can see. There are different kinds of proteins that are present. So the proteins are found in keratin, in our hair, nails, or in the horns and claws of animals or birds. And the collagen, it's in our skin, in our ligaments, cartilage, hemoglobin in our blood. We have muscle, that the muscle is formed up of proteins which will help in the movement and heart functions. We have antibodies that fight off the infection in bacteria, that is also a kind of protein. And most importantly, the membrane channels, these are the channels that allow substances in and out of the cells. Those are also formed of the protein. So here we can see that how important the proteins are in human body, not just in human body but in different life forms as well. So here we end up with the introduction of the proteins. In case there is any other topic that you need an explanation for, you can always write to us. Your feedback is most welcome. So looking forward for your feedback. Here we end the video. Thank you so much.